my question is for Dr. Pim. In your lab, did you transform the fibroblast into myoblast, and then did you try to inject this myoblast into mice model of Pompe disease, for example, and seeing the result?
Yeah, so this is what we were and actually doing right.
You didn't do it yet?
Doesn't work? Yeah. So what we actually aim to do is to transplant these cells. And see if we can contribute to muscle formation in a mouse. And you have to. These are human cells, and if you transplant them in a mouse, you need to prevent that the human cells are attacked. So we use an immunodeficient mouse that prevents a mouse model actually for Pompe disease. So that we have made. But these experiments are not very easy, and but this is our next aim to do. Yeah.
Okay, thank you.
Has ever myoblast transplant been successful?
So you have to make a distinction between the clinical trials where. So in earlier years, people have tried to transplant myoblasts, but myoblasts are differentiated cells, so they are not stem cells. And these have completely failed, these clinical trials. So that's not the way to go. So what you need to have is a cell that is not so differentiated into the myogenic lineage, and so therefore it's very exciting that we can generate muscle stem cells from iPS cells. And there the hope is that this will provide a source, and that actually has been shown in some work that has been published. The group of Rita Pellinghero has used inducible overexpression of PAC7 to create the cells that I just showed. Of course, then you have an extra gene, which is a complication, but she showed that you can transplant these cells in a mouse and that they will contribute to muscle formation. So in principle, hopefully it is possible.
I just make a short comment on this. So there's ongoing in a few limb-girdle muscular dystrophies also this approach done by a group in London. And of course they show that this is possible. So it's very important, your work, what you're doing now with the stem cells, because that can work, the myoblast transfer. So that was back to Karpati's work in the 70s, starting in the 70s, was no way to do it, unfortunately.
Lebowicz. Hi, question for Dr. Raben. So your compound X is very interesting, and if I understood correctly, at low dose it promotes exocytosis, and at higher dose TFEB or TFE3 nuclear translocation. Can we then infer that the mechanism of exocytosis does not depend on TFEB or TFE3 nuclear translocation, or no?
No. I probably didn't make it clear. Both at low and high dosages, it stimulates lysosomal exocytosis, and its activation of endogenous transcription factor, and its translocation to the nucleus. At low dosage, what I showed was that there is in the nucleus, so you can see TFE3 in the nucleus, but you can see the effect of it, the realignment of the lysosome. Essentially, it's the same thing. Higher dosage, you see more in the nucleus, and perhaps more lysosomal exocytosis.
I actually had two questions. One for you, Dr. Raben. How would you utilize the acid nanoparticles? Like, how would you implement them?
These nanoparticles are available. They are certainly now for experimental use, so you can buy them. We have a collaboration with people who are involved in nanoparticles, and they are actually developing nanoparticles for commercial use for liver. So we hope to be able to stimulate them to generate another one if we can show that in tissue culture it really works. So this is very much doable. It's
for
probably easier to deliver to muscle than enzyme for enzyme replacement therapy. They're tiny.
And the other is for Dr. Pijnappel. My experience in the past has been with bone marrow transplant and stem cell transplant for oncology. So my question is, utilization of the stem cells or the satellite cells that you're referring to, would it be administered in the same way that a bone marrow transplant or a stem cell transplant is done for cancer by ablating and then re-administering intravenously, or do you have another methodology for that?
Yeah, that's an important point. So when we do this in a mouse, we injure the mouse one day before, and that triggers the regeneration system, and then you give the satellite cells a chance to be there and to grow and to make muscle. So when you will do this in human patients or testis, you probably have to trigger the muscle to get the cells somehow. It's not like chemotherapy that you use in bone marrow transplantations, but what we are thinking of is mild injury. For instance, exercise could be one option where you will damage the muscle a little bit, and then it will be triggered to take up other cells. But this is an important point. And in a mouse, what we use are toxins, and there's also actually a milder toxin that is that can also injure the muscle, and it has been approved for human use. It's already used in a clinic. So, but it's important that you have to condition the muscle. Yes.
Thank you.
I have a question over there. Yes, please.
Okay. I have a question for Dr. Raben. So you presented very compelling evidence that autophagy was implicated in the in the pathogenesis of Pompe disease. Can you hear me? Okay, hi.
Yes.
So a question for Dr. Raben regarding autophagy as being an important pathogenic marker in the disease. So what is your experience of modifiers like treatments that upregulate or downregulate in the mouse? And I would love to hear experience in patients if they have tried this.
It's such a long story. Okay.
Very short.
So, yes, okay, I'll try to make it short. What we see is an induction of autophagy. So autophagy is a process, has a beginning and an end. Beginning is a production of autophagosome. The end is the resolution in the lysosome. So we have both ends of the process impaired, increased induction and decreased resolution. So essentially, what you can do is you can suppress autophagy, which we did genetically in a mouse, and showed that then the enzyme replacement therapy worked very well. Or you can stimulate the fusion of autophagosomes and lysosomes. I can give you an idea. So it's a traffic jam. What you can do, you can stop, you can reduce the number of the cars, or you can open the gates. And so both approaches are very reasonable. And by the way, now, especially in cancer field, there are multiple inhibitors of autophagy, which would be very reasonable to try. We don't want to genetically suppress it, but it would be reasonable to try the inhibitors of autophagy in combination with enzyme replacement therapy. Or the other approach, transcription factor, which would still open the gate. So both make sense.
Excuse me, just the microphone.
Tried this in patients?
You heard it?
No, I didn't.
I haven't heard that.
Has anyone?
Tried any of these modifiers in patients with
The modifiers. This is about modifiers. Anyone tried modifiers?
rapamycin or chloroquine or anything else that's worked in autophagy?
Oh, chloroquine wouldn't work because it leads to excretion of acetyl-glucosidase. So even the little bit of endogenous activity will be lost.
What was the question?
Chloroquine. Rapamycin, I think people are studying rapamycin because it affects the
No.
tuberous sclerosis pathway. No?
Because rapamycin...
That's important.
Yes. Rapamycin is mTOR inhibitor. mTOR inhibits autophagy. With rapamycin, you would stimulate autophagy.
So no rapamycin in Pompe disease?
No.
You agree, Giancarlo?
No, I'm not aware of that. That's
But
not
from my
I
perspective,
think there was
no.
something in the literature. Is there anyone that want to comment on rapamycin? Rapamycin out?
Out.
Rapamycin. There was another question over there.
Yeah, quick comment. The Genzyme group actually evaluated rapamycin in the GA knockout mice, and it had beneficial effects by suppressing glycogen synthase, but it was a very small experiment, and that was an unexpected effect.
Right. Yes.
I also have a question
So
for...
I'm certainly very familiar with this paper. They claim that there is a reduction in the level of glycogen without affecting autophagy. And there was another paper which pretty much contradicts it. So yes, this needs to be clarified.
Quick question for Dr. Danesino, thank you. So the ACE polymorphism activates ACE activity. It's detrimental in Pompe disease. So could you speculate about the effect of an ACE inhibitor? Could that be beneficial? Thank you.
Okay. In theory, it should be possible. I think there was a very small study, again, on McArdle disease, trying to attempt it, but then I haven't seen any further study on it. Of course, the amount of blood supply to tissues, which may be partially regulated by the levels of AC, could be relevant for building up muscle and maybe also for the response of therapy. It could be something that could be studied in the future.
I have a comment about the rapamycin. Similar to what Nina mentions, we had done it in glycogen storage disease type 3, and it does suppress glycogen synthase, but it does upregulate autophagy. So we had the same results as what you mentioned, Nina. But this was in GST type 3. It's dysregulation. No, the glycogen synthase is good, but the dysregulation of autophagy, which was seen, is not helpful.
So you agree too that rapamycin is out?
Correct.
Okay, that's handy. Is it on?
Hello. Yes, I have a question, and I think this question is just for maybe, I'm hoping that I'm not alone. Of all the things, I mean, it's very admirable that you guys are doing all of this in treating the muscle, the injured or the lost muscle, the damaged muscles. But my question is the other way. I want to know if there's any effort in trying to find or to create the defective genes that makes the enzyme, rather than treating, but to make the enzyme to help the people with the missing enzyme that is causing all of this problem. Thank you.
Yeah, I think maybe I'll answer the question. There is shortage of enzyme. So, I mean, enzyme replacement therapy tries to raise the amount of enzyme to solve the problem. And the gene therapy approaches that we heard about yesterday, you try to provide correct genetic information to increase the acid alpha glucosidase production. So those are the two solutions. But maybe that does not answer your question.
Well, yeah, that's partially addressed it, but not completely giving me the very simple question. Is that when you knock out, when you injured or whatever to the mouse, to make the mouse is a Pompe candidate, you did something to the mouse. You render something that the mouse no longer make the enzyme.
Right, yes.
Can you,
Okay, yeah.
tell
I
us-
got it.
You
I
got
got it.
Thank you.
So yeah, I got it. So how do you make a mouse with Pompe disease? Both of us did it, and you manipulate actually the gene, but you do it outside the mouse. So you start with a fertilized mouse oocyte, and you manipulate a human embryo, or a mouse embryo in this case, that you grow up, and then from the embryo you grow a mouse, which is lacking the acid alpha-glucosidase gene. So that approach, how did you manipulate, how did you do it? You took a mouse embryo, a single cell mouse embryo, and you manipulate it in the laboratory, and from that you've built a mouse. So in the human situation, that would be impossible. To do it in the human situation, you're looking for a drug or a small molecules that would be able to find in every cell of the body, and then we are talking about billions and billions of cells, a drug that would go into these cells, integrate somehow into the genetic information, find the acid alpha-glucosidase gene among the 25,000, let's say that, among the 25,000 genes, and then adhere to it, and by DNA repair processes, not excluded, fix the defect. But that's far future, but maybe not that far. You never know. I think that's your answer. Eh? Fix the problem in the DNA.
Okay, I'm sorry for that. I understand what Mrs. LeBrown wanted to know about. I think it's a question that all the parents, grandparents, all the patients want to know. Is it possible to cure it? And I will come with something that makes me think about correcting the DNA. I don't know if I'm correct. There's in Florida, they have a clinical trial for a kind of disease that, like a baby who doesn't have any immunitary system. A bubble baby, exactly. And it's a problem that comes from the DNA. And they did cure the baby. When in my country, a Montreal baby went there and came back a year and a half after, cured. And I was thinking, oh my God, they did it. It's something that they've been trying for so long. And that made me think, the same question of you, Mrs. Brown, is it possible to have it with the GAA gene? I know it's maybe far, far away, but what's the difference? My question, finally, is what's the difference between curing that bubble baby with the DNA problem?
All right, the bubble baby is a problem with the blood cells. There's something wrong with the cells in the blood. So only the cells in the blood and not the entire body. And what you do, you transplant this baby with bone marrow stem cells. So you take out the stem cells from the baby that don't produce this enzyme, it's also a protein, and then you replace it by a normal bone marrow stem cell. And this normal bone marrow stem cell then starts to populate the whole circulation, and then you fix it. But this is the approach that was reported yesterday in the gene, that you take bone marrow from a patient, you manipulate outside the body, so you introduce with a virus an absolutely normal human acid alpha-glucosidase gene, and then you transplant the bone marrow cells, and the bone marrow cells will populate the cells in the blood, and if they produce acid alpha-glucosidase in high enough amount, then they can serve as a source. Okay, once you have understood this, then you will hear Dr. Raben doesn't agree. Okay, now with you.
problem in Pompe disease that those cells need to.
Yeah, right. Yeah, so the enzyme then has to come from the white blood cells and then go to the muscles the same way as you have enzyme replacement therapy. So you need extremely high levels of production because otherwise it doesn't work. That's your point, Nina? Pim?
So the whole idea is that if you, so you can use blood cells, but you can also use stem cells that I just showed, and this is the idea. So if you would try cells that you correct, then you have the same goal. Right. But this is very difficult. So, but yeah.
Yeah, you want a comment?
Yeah. No, at the end, I think with lentiviral factors, you have there's now a trial also very soon for SCID. That is the severe combined immune deficiency. That's the bubble baby, and that's also done with lentiviral. And I think the positive things is that these cells, these blood cells, move to all types of tissues and also to the brain. And also if you can have the factories over there, so it's not only in the liver, but also in other cells, and maybe also in the muscle, although it's very difficult to get blood cells in the muscle, but there you have the factory at the spot where you want to have the enzyme. So that's the advantage. So I also heard another very positive story, then I go to another subject, Nina. Because what you showed is that in the myoblast, in your tissue culture disc, you didn't see the problem with the TFAP and the consequences of TFE3. And isn't also that not a very positive message for patients with for the enzyme replacement therapy? Apparently, if you're early enough and if there is not already the consequences of autophagic buildup, that you can prevent this autophagic buildup. So that, in fact, it's a plea that you should be there with enzyme replacement therapy before the autophagic buildup has happened. Is that the message?
All the experiments in vitro, in fibroblasts, in myoblasts, they work beautifully with enzyme replacement therapy, right? So, because these are the early stages. That's absolutely right, yes.
I have a question for him. And in Italy, we talk a lot and with a lot of confusion about the stem cell infusion. And a lot of people of our group of patients thinks that with an infusion of stem cell, he can be better. But I think no. So I want to understand the really things about stem cell infusion for our disease.
I'm not sure which stem cell infusions, which stem cells they use you are referring to, but I can tell you that at this point it's not really possible yet, but this is what we are working on. So we are really, or Henrike knows maybe?
Are you talking about a normal stem cell transfusion? So that means without the lentiviral modification? I think you are. Could you answer that?
Sorry, yeah, thanks. So the problem there, that's a bone marrow transplantation you're referring to probably. So there the point is that you have to imagine the picture that the bone marrow has stem cells, blood stem cells, and they are going to produce cells for the blood, and they are going to be in your whole body. The idea, what I talked about yesterday, is we put a virus with the GAA gene, and we do that to have more of the same gene. If you don't do that, like you're talking about, then the level of the gene, of the protein that's made from the gene, is too low. And that's the problem. So we know that for Pompe disease, it's not enough to do that. So you have to imagine that, of course, the cells will produce GAA protein. The protein, a little bit of that comes out and will be taken up by muscle cells. But just the amount is just too low.
I think the bone marrow transplantation has been tried before and it didn't work. So that's the reason why you need a modification of the stem cells to enable them to have a higher production of your enzyme. So a normal stem cell transplantation will not work.
Thanks, very clear. There's one last question, and then we go for a coffee break. Yes, please.
So, in trying to understand all this, with the new information about the autophagy and the mTOR-C and all the other things you folks talked about, does this mean that there is something else besides a lack of GAA going on, or these are just additional effects of the GAA that weren't realized before?
Okay. Yeah, it's better. So the GAA defect results in what we call the primary, this is the primary defect. This is the accumulation of glycogen in the lysosomes is the primary and the major effect of the enzyme deficiency. Once lysosomes themselves become enlarged and filled with glycogen, this initiates the whole cascade of the secondary events resulting from the enlargement of lysosome. So essentially, lysosomes become much less functional. And a lot of the pathways and a lot of the processes in the cell depend on the normal functioning of the lysosomes. So these are what we call a secondary event, the whole cascade of the secondary events. Okay, thank you everybody. Thank you very much.
Thanks for all the questions.
Yes, thank you for all the questions.
All right, so we have 5 companies, and we love them all equally well, and we don't want them to fight, so they will present in alphabetical order. So Amicus is first, and the presentation will be by Dr. Nina Patel, and she is Director, Patient and Professional Advocacy at Amicus. The floor is yours, and the mic is
on.
Yeah.
Hello, can you hear me?
Is it on?
Is it on? Yeah. Right. Hello, everybody. It's a great privilege to be here, and I would really like to thank AMDA and International Pompe Association for inviting Amicus to speak. Congratulations to AMDA for their 20 years. What a great milestones, and we thank you for all the work and support you have given to the community. My name is Nita Patel. Although I'm not a doctor, I'm a nurse. I'm a genetic nurse with a lot of experience in lysosomal storage disorders, and most of my knowledge comes from patients. Amicus Therapeutics, this is our mission statement. Yep. And I wanted to just let you know that currently we do not have a product on the market. We're doing clinical research in many disorders, mainly lysosomal storage disorders. And as you can see on the slide here, on the left-hand side, you see a couple of siblings with Pompe disease. We are doing some research in Pompe. In the center is Fabry. It's a family with Fabry disease. That's our lead program. And on the right, it's our newest addition. We're working in a condition known as epidermolysis bullosa, another very rare and devastating disease. So Amicus is a very small company. We were headquartered in Cranberry, which is where our headquarters are currently. However, in the recent past, we have really grown quite a bit. We do have international headquarters now in the UK, and I think within the next couple of weeks, our Gerrards Cross office is going to open. We also have offices in Germany, France, and Netherlands, as well as consultants working globally. what does patient advocacy do? And I actually want to ask the audience here, if you're a patient organization leader, could you just please put your hand up? There are so many patient organization leaders, and I just want us to applaud their efforts in supporting the community, the professional and the patient community. They go out of their way to learn about the disease and support the areas and geographic areas where the patients are, and I think they just do an amazing job. Can we please just give them an applause? I also want to say that at Amicus and patient advocacy, we obviously work with the patient organization leaders, but we also, because of the company CEO that's quite well recognized, we get a lot of questions in patient advocacy that come to us, not just from Pompe, Fabre, EB now, from anybody. And there are patients who have recently been diagnosed with whatever disease that they have, and they just are looking for help. And in patient advocacy, obviously we're not going to turn them away, but patient organization leaders like yourself in all the diseases do help, and we connect the patients with the patient organization leaders and the communities and the umbrella organizations so that they can get the need and their needs met, which is they're not alone in anything. Any patient is not alone. There's always somebody out there that can help you. We collaborate with all stakeholders, patient, the physicians here are just amazing. I have worked as a nurse since I was 18, and I tell you, the physicians in rare diseases are very, very special. They're almost like the physicians that, in the olden days where they used to have the little bags and go to your homes and give you personalized care. The physicians I have met are so dedicated to their patient population and so protective of them, and they really deserve an applause too. They're just great. Thank you. We just go around applauding everybody today. So obviously we work with all these folks that are here. We provide and learn from you guys whatever your needs are and bring them back to the company. And that's what we do internally. Patient organizations and patient advocacies, I come to meetings like this, learn what your unmet needs are, and then bring them back to the bench scientists, the administrative staff at Amicus, because they need to know what the patients are going through and what their work is really for, to keep them engaged in trying to do the best work they do. And that's what my goal is to connect the two and be the conduit of providing education. This is again, just this slide is telling you how we do this, how patient advocacy and how we work with the community. We do have a patient advisory board. So, companies have medical advisory boards, they'll have a scientific advisory board, and there are experts that come to the table and actually discuss ideas and how to develop a protocol or a clinical trial. We feel that patients also need to have a voice at that table, and we have a patient advisory board. These are informed persons affected with, in this case, Pompe disease, representing their self or their community. It's the patients have to go through an application and nomination process. Each patient advisory board member has a commitment of a minimum of 2 years. And then if they want to leave, then we will replace them. We meet at least once or twice a year, depending on the needs, either of our needs or the patient's needs. And we actually have got so much information from our patient advisory board. You have a picture of the advisory board members. We met at World last year in Florida. And we had a meeting there, a full-day meeting, and it was very, very informative, and I want to thank them. We do this. Patient advisory boards are subject to Amicus's policies and procedures relating to compliance with the FDA and healthcare laws, and we try and collaborate together with the whole advisory board. these are some of the patient organization that we collaborate with. We speak with the leaders there. We learn geographically what each area's unmet needs are, and we bring that back to Amicus so that when we develop our clinical trials and our protocols, we are addressing the needs globally and not just in the US. I just wanted to say that although I'm not sharing with you what we are doing in our research, we are currently working on a new enzyme replacement therapy in combination with pharmacological chaperones. It's going to be in the clinic hopefully next year, and you can get more information about this from the physicians. We do speak with our physicians and they are aware of it. You can get information from patient organization leaders, and this trial, when it is available, will be listed on clinicaltrials.gov. If you do have questions, and if I'm able to help in any way, you know, the three patient advocacy folks at Amicus are listed on the slide there. Jane Gershkowitz is our chief patient advocate. I work with her, and of course I'm the US person. And we have a very new head of international patient advocacy, Jenny Wilson, who has quite a lot of experience with rare diseases and is, as I've learned, is a wonderful person to work with. So I'm going to keep this short. I'll give some time back to you guys. If you have any questions, please feel free to email me, and thank you very much.
Thank you very much for your presentation. You stayed within time. I appreciate it. Next speaker, Dr. Suyash Prasad. Oh, there he is. Hi, Suyash. Audentes these days, isn't it?
Yes, that's correct. These days, I believe. Let me just click that. Down to the right. There we go. To that one. This one. Thank you. Great.
I had this problem yesterday. That's why I learned.
So, good morning, everybody. Very pleased to be here. Yes, my name is Suyash Prasad, and I'm a paediatrician by training, and worked for a number of years in clinical practice in London and in Sydney, and then moved to industry about 15 years ago, where I worked at a number of companies, including Genzyme and Biomarin, which is the reason for Arnold's comment. And most recently was asked to join Audentes about 18 months ago as the Chief Medical Officer. So I'm here to tell you a little bit about Audentes. We're a very new, very small company. I'll tell you a little bit about us as a company, tell you a little bit about our pipeline. Our focus is gene therapy, and I'll speak a little bit about how is a gene therapy different to a drug. You heard a little bit about this yesterday from the gene therapy workshop. I'm going to talk a bit about clinical trials, what we need to do before we start clinical trials, and also how you as families and children and adults affected by Pompe can help us. So the story of Audentes, and you can hear me okay, yes? Yeah. Story of Audentes. The company was started about 2 years ago by our CEO, Matt Patterson. Now, Audentes is a Latin word which means to have boldness or to have courage. And the reason Matt selected this name is because he's worked for a number of years in rare diseases, actually for more companies than I have. He's worked at Amicus, Genzyme and Biomarin. And he was always impressed by the courage that families and children affected by rare diseases have displayed, which is one of the reasons for the name of the company. The other reason is that we need bold, innovative scientific solutions, which is why he chose the name Audentes, Latin for to have courageousness. The other reason, which he doesn't mention very often, is that his mother's a Latin teacher, and I think it's, partly a homage to her. Maybe there's some mother-son guilt issues, I'm not entirely sure. But That's the other reason. So he raised some seed funding about 2 years ago, and since then we've had 3 successive rounds over 2 years of financing. So we're a privately held company. We have last round of financing completed about 2 months ago. There's a press release about it. And we have predominantly venture capital funding, but a lot of large banks have also invested in us. And for our size, it's fine. At some point in the future, when we get bigger, we'll probably have to become a publicly traded company, depending on how things progress over the next period of time. We're based in San Francisco. I was the 8th employee when I joined 18 months ago. We now have 38 employees. We have laboratory space in San Francisco, and we also are developing our own in-house manufacturing facility. So we have an orphan disease focus, we have a gene therapy focus, specifically AAV gene therapy. We're hoping for some of our programs to enter clinical trials during the course of the next couple of years. We have an expanding portfolio. We have 3 programs in our portfolio, and we're building our own manufacturing capabilities also. Because we realize that with a lot of these biologicals, the manufacturing side of things can be challenging. There's been several experiences over the past few years where we realize it's important to really take that into our own hands and make sure we have our own reliable manufacturing facilities to supply clinical trials and eventual commercial launch of products. We have an experienced leadership team, and we have a group of high-quality investors who have a track record in investing in a number of small startup biotechnology companies. So, we have a number of us at this meeting. Hopefully you've met some of us so far. If you haven't, then here are their pictures. We want you to come and say hello to us. In the top right-hand corner, we have Barbara Webbles, who is our Vice President of Patient Advocacy. She is over in the far corner over there, so she's waving now. So over lunch, please go and say hello to Barbara. If you're interested in staying in touch with Audentes, give her your details or subscribe to our website, and we'll send you some updates. We have Jeff, who is our Vice President of Clinical Development, another senior physician. Magda, who heads up medical writing. She's involved in, she's over there. Wave again, Magda. Great. Miko, who heads up regulatory affairs, also sitting next to Magda, and then Sarah, who runs our clinical trials. So over lunch, please come and say hello to us. Tell us what you think about the presentation. Tell us what you think about Audentes, and tell us a bit about your lives. We're really here to learn from you. So we have 3 programs in development, one of which is Pompe. And you heard the beginnings of this yesterday. There was a gene therapy session. Barry Burns spoke, Dwight Coble spoke, Pim Pijnappel spoke, and Andrea Amalfitano spoke. We're actually partnering with Barry Byrne on the AAV9 project. We've acquired the rights to that technology, and based on Barry's initial work, which he shared yesterday, our responsibility here is to take this through IND-enabling animal studies, i.e. complete dose-finding studies in the mouse model, complete toxicology studies in the non-human primates, and then start clinical development. We have 2 other programs. We have a program, an AAV8 program in another disease called X-linked myotubular myopathy, and an AAV9 program in, I always have to take a deep breath before I say this, catecholaminergic polymorphic ventricular tachycardia. So, or CPVT. I thought I'd get a round of applause, but clearly not. CPVT, I had to spend a few days learning. Thank you. And we're actually looking at bringing on additional gene therapy programs as the company grows. So CPVT, let me tell you a bit about this. It's a cardiac disease of sudden death in the young. You often hear stories of, a teenage boy running out on the football field and collapsing and then dying with a cardiac problem, or a young girl going to a party, feeling unwell, and then collapsing and dying. And there are a number of syndromes that cause this cardiac disease of sudden death in the young. CPVT is one of those. It's a malignant inherited arrhythmogenic disease caused by abnormal calcium homeostasis. The prevalence is estimated at about 6,000, so it does fit within the categories of rare disease. Is that okay? No? Okay.
No, it doesn't sound okay.
Okay, great. Thank you. That better? Okay. Disease is characterized by high unmet need, and we have some positive proof of concept data in the mouse model of this disease. And so, once again, our plan here is to continue with some IND-enabling animal studies and start a program for clinical development for that particular product. Our other program is X-linked myotubular myopathy, and I wanted to share a bit of information on this because there are many parallels between this disease and Pompe disease. It's a genetic disease characterized by extreme muscle weakness, respiratory failure, and early death. The mutation is in the MTM1 gene. There is a deficiency of a protein called myotubularin. It's an enzyme which allows skeletal muscles to contract. In the absence of this enzyme, the children can't move. So when they're born, they are hypotonic, they're floppy, they can't breathe for themselves, they need intubation and ventilation and urgent management. There's a high mortality rate. 50% or so die, up to 50% die within the first year. Sometimes clinicians advise the families that there's nothing that can be done and allow the patient to die. There are some that survive, and of those that do survive, past infancy, they require ventilator support, feeding tubes, often can't walk. And you can see in some of the pictures, you can see the myopathy, the hypertonic faces, the ptosis, the open mouth, the supports to prevent development of scoliosis, contractures, the tracheostomy, the ventilator. And so many parallels with Pompe. We have positive data in the mouse model and the dog model for this using an AAV8 gene therapy approach, and we're hoping to start clinical trials also of this program once we've completed all the necessary toxicology studies in the animal model. One of the things we like to do is talk to patients and families, and we've had a number of advisory boards, advisory groups. We had one recently for Pompe disease just a few weeks ago, and we're just compiling and synthesizing this information. But I thought I'd share some of the learnings from the MTM patient focus groups. I think there's many parallels between that condition and Pompe, and some of this may resonate. We had two meetings, one in Europe in London and the other one in Chicago. And, we learned many, many things, but here are some of the highlights from those discussions. We learned that MTM is a severe and devastating illness, not surprisingly, has a major impact on the well-being of families. In line with other rare diseases, the diagnostic process is not as smooth as one would hope. Some parents had to actively push to get a muscle biopsy. Some parents had to push to prevent withdrawal of care or for referral to an expert centre. There are social burdens. Those that survive, children have few friends. There's concerns about exposure to children due to infection risk. Parents have to have extended, frequent time off work. There's a huge psychological, emotional burden. There are physical burdens, wheelchair use, ventilators. There are concerns about battery life, electrical sources, worries of failure. We've met a couple of children with MTM whose ventilators have failed through the night and the children have died. There are limited transport options available. Modifications need to be made to houses. One of the interesting things is that there's actually less support available, interestingly, for families and patients in the US than there is in Europe. In the UK, for example, the families are given a grant of £30,000, so about $50,000, to modify houses to make them better equipped for children with such challenges. In Germany, 24-hour nursing care is provided by the state, so it's kind of an interesting difference. Medical concerns about ventilator use, risks of chest infections, etc. And, when we asked the question, what results would you like to see in your child from a potential therapy, these are some of the answers we heard. We heard that parents would like to be able to put their child in the car, disconnect them from the ventilator for a few moments to be able to do that. Have them be more robust, be independent from machines, be able to communicate, breathe independently, swallow. One mother wanted to be able to sing with her child. Even just being able to reposition themselves. And these are important insights that we as physicians and we as companies need to hear. I think Hans van der Ploeg yesterday made the comment that it's important for clinicians to understand the patient perspective, and she used the word precious. It's precious for us to understand it's important for us to understand what's precious to you as families, because this helps us in our discussions with regulators. When we go to the FDA or the MHRA and say, look, we've spoken to families and it's important from a family perspective, what's important to them is the respiratory aspect of the disease, which is why we've selected the mean inspiratory pressure as an endpoint. They understand that kind of language. So we need to keep learning from you. So please do keep talking with us, keep sharing with us. be involved in these advisory boards that several of the companies set up and run. It's very helpful for us. Okay, I won't touch on gene therapies too much. You heard a lot about this yesterday, but basically, enzymes are protein molecules. Proteins have specific functions. If you lack the protein, then there is a function that is lacking in the body or in the organ. Thanks, Arnold. Often the absence of this enzyme is due to a genetic defect, and that means there's a piece of DNA that's missing. So how do you treat this lack of functioning due to an enzyme deficit? Well, you can give a drug that helps whatever enzyme that is present to function more optimally. This is the basis behind the chaperone approach. You can synthesize the enzyme out of the body and then replace it. That's enzyme replacement therapy. Or you can take a gene therapy approach, where you replace the gene that's missing and hope that gene produces the enzyme that's lacking within the body, which is the gene therapy approach. So what we anticipate happening is that there will be a solution of virus particles, and these are large, large numbers of virus particles. You're talking about 10 to the power 12, 10 to the power 13 vector genomes per kilo. So billions and billions of virus particles being injected into the body, which then carry the DNA to all the cells within the body. The virus particles enter the cells, they unload the gene, and the gene starts producing the missing protein or the missing enzyme. All sounds simple, but it's far more complex than that. But that's the theory behind the gene therapy approach. We obviously need to run clinical trials, and one of the questions yesterday was really about, how soon do we have to wait? How long do we have to wait before this starts clinical trials, before these therapies get into the clinic, before they are actually available to help our children and families? Well, it takes a long time. Barry Byrne mentioned yesterday that the one gene therapy product that's approved in Europe, there's no gene therapies approved in the US, is a drug. He didn't mention the name. The drug is called Libera. It's for a disease called lipoprotein acid lipase deficiency. It was approved in 2012, but development started in 1996, so it took more than 15 years. So we're talking a long, long time. You've got to go through this whole process: animal studies, proof of concept studies in humans, larger clinical trials in humans, file the data with the authorities, get the approval. All the while, you are having to manufacture increasing amounts of drug. Now, I'm being told we have a couple of minutes, so very briefly, what needs to happen before a clinical trial in patients can occur? We need to make sure we can manufacture the drug, make enough product, the production system is reliable, the production suite is fully sterile and controlled. We need to learn what we can from the animal data. We're fortunate in Pompe that we have a good, well-understood knockout mouse model. And preliminary data in this knockout mouse model, which Barry shared a bit of yesterday, suggests that the AAV9 gene therapy approach works quite nicely, but there are many, many differences between the knockout mouse and the human. So there's many more animal studies that need to be completed. We need to design the clinical trial. We need to make sure the trial can be run operationally. We have to select sites. Which centers? Have centers run gene therapy studies before? Have they run rare disease studies before? Are they used to processing samples of tissue? And muscle biopsies in gene therapy studies for Pompe will be far more complex. You won't just look at glycogen, you'll need to also look at things like vector genome copy number, mRNA, DNA. What scientific decisions need to be made? And the first question here is the important one: what is the dose to be selected? Once you've given the gene therapy product, you can't take it away. It's not like an enzyme where you can stop the infusion halfway through. Once it's given, it's given. And you need to therefore make sure you get the dose right in case there's an adverse event. How many patients? Will there be a placebo control? Inclusion criteria? What safety monitoring? Many more decisions need to be made. So where are we now? Where are Audentes now? Well, we're planning to talk with regulatory agencies about the current program in Europe and the US. Once we've had these discussions, we'll be able to share more information about what our trial might look like. We're talking with experts internationally. We're talking with the patient community. Likely we'll have another patient advisory board or focus group in the near future. And we're performing the IND-enabling animal studies and manufacturing planning. And we're doing some further laboratory research, vector design, vector optimization, promoter design. How can you contribute? You can take part in research, talk to other families and children, visit the Audentes patient website, support the charitable organizations. Attendance at meetings such as these where you're educating yourselves is very important. And in summary, therefore, hopefully we've shared a little bit about the organisation, talked a bit about how gene therapy is different to a drug, what needs to happen before we start clinical trials. Please come and say hello to us. I'm looking forward to being able to come and share more information with you on an ongoing basis, and look forward to a long and ongoing engagement. Thank you.
Thank you too for this very clear presentation. And the next speaker is Dr. Liron Walsh from Biomarin. Hi. You can open it yourself. You were cert with this one, I guess.
Yeah.
Want to click it open?
Sure. Yeah,
perfect.
Go. Hi, good morning, everybody. I'm Liron Walsh, the medical director for the Pompe program. It's really an honor and a privilege to be presenting in front of all of you, not just on behalf of Biomarin, but on behalf of the investigators and all the patients that have taken part in our clinical program to date. Over the last few days, it's been really inspiring for me, actually, first time at this meeting, to hear many of your stories and some of your setbacks, but also the triumphs, and really your dedication to bringing new therapies to Pompe. And really that's one of my goals as well. And in joining Biomarin, I joined a company that has 5 marketed products already, 11 in the pipeline, really grow a global infrastructure to conduct clinical studies. And so I'm pleased to present the revoglucosidase alpha clinical program to you. As many of you heard over the last several days, the importance of respiratory function, and particularly the diaphragm is that critical muscle of respiration. And we saw Dr. Smith show some videos the other day about how the diaphragm doesn't move as well in Pompe patients. And in fact, over time, as the disease progresses, certainly without treatment, the muscle weakens and leads to low lung volumes, impaired cough, and sleep-disordered breathing, which can lead to fatigue, headaches, and just not feeling well throughout the day. The inspiratory muscle strength actually can predict the onset of some of these things, such as ventilatory support, need for ventilatory support, sleep-disordered breathing, and wheelchair use. And so how do we measure respiratory muscle strength in the clinic? So the one way to do it is through the transdiaphragmatic way that Dr. Smith discussed yesterday with the phrenic nerve stimulation, but that's obviously not so practical. But there are machines, these small handheld devices, where probably many of you have used it in your clinics, and one can use this sort of handheld device, take a nice large inspiratory breath, and there's a value there, and you can follow that over time. It's not an easy test to do. I've done it myself, and it takes some training, but with appropriate training for patients and for clinic staff, it's one that is quite valuable, particularly in following patients over time. And there are other studies in other disease states, such as ALS, where this can be used to follow patients. So while Biomarin program has a focus on respiratory muscle strength, revoglucosidase alpha, I sometimes get that wrong, is a potential next generation enzyme replacement therapy. But the idea is to deliver enzyme not just to the diaphragm, but to globally to muscles throughout the body. Revoglucosidase alpha is a GAA, the active enzyme, which is tagged to IGF-2, insulin-like growth factor. And we discussed this a little bit yesterday from the group from Rotterdam. And it's designed to help the enzyme reach the lysosome. It binds with very high affinity to the receptors on the muscle cell, which allows it to be taken up and delivered to the lysosome for hopefully effective treatment. It's just a little cartoon of kind of what it looks like binding to the receptor and trafficking to the lysosome. Mouse models and in vitro data supported the use of this product for initiation of phase 1 and phase 2 clinical studies. The phase 1, phase 2 clinical study, which was an open label safety and efficacy study in treatment naive, so those that have not had ERT before, began in 2011. So we have quite a lot of experience so far with that drug in treatment naive patients. There were 3 dose cohorts enrolled, 3 patients at 5, 3 patients at 10, and 16 patients at 20 milligrams. Today, all but 1 patient is on the 20 milligrams per kilogram every other week. The enrollment criteria were fairly standard for a phase 1, phase 2 study. Late onset Pompe disease, naive to prior therapy, ability to walk at least 40 meters with or without a walking aid, and some respiratory impairment as defined by a low FVC or a decrease when moving from sitting to lying down. Patients were followed for 24 weeks and had the opportunity to enroll into an extension phase. I'm going to present the safety profile first, given that this was really the goal of this phase 1/2 study. And I'll present to you the 72-week data. Generally speaking, the enzyme was well tolerated. Adverse events, other than reductions in blood glucose, which I'll speak about in a moment, were similar to those seen with really other ERTs. Reductions in blood glucose are a pharmacological effect of the tag. It's an insulin-like growth factor. So for those that are probably familiar with insulin, insulin acts to reduce blood glucose. For the vast majority of patients, these are really mild reductions. And as of probably the summertime, we've done 1,000 infusions in this phase 1/2 study, and there are only really a handful of cases where the reduction in glucose would be considered of significance. Most patients can easily treat reductions in glucose by a snack, in some cases an infusion of glucose during the infusion of the enzyme. For the most part, they are manageable, and we've really learned a lot from the patients that have been on study in that now we're asking patients to have an appropriate, well-balanced meal prior to the infusion and snacks throughout in order to maintain blood glucose in a safe level. I think importantly is we found improvements in respiratory muscle strength, and those are maintained through 72 weeks. The y-axis here looks at the change from baseline and maximum inspiratory pressure. That's patients using that handheld device. And you could see that there's an initial increase to about 14 points in percent predicted, which is maintained over the 72 weeks of treatment. There was also an improvement in 6-minute walk test. I'm going to point to the other side here. I feel bad, but this side always gets my laser pointer. Oh, or the cursor is a better idea. Here we go. Yeah, I got it. That works, sort of. All right, works for you, works for me. So we had an increase of about 24 meters over the course of 72 weeks. I should mention that 6-minute walk test improvements may also be related to improvements in respiratory muscle strength given some measure of endurance, but we do have a subset of patients that had muscle biopsies, and those muscle biopsies actually also show reduction in glycogen. FVC had a slight increase of about 2 points in percent predicted with some trailing off, but overall FVCs, this is hard to use. There we go. Overall, FVC is maintained over the 72 weeks of treatment. With these compelling results, at least we felt so, that they were compelling, we initiated a phase 3 program. So the phase 1/2 study, which demonstrated acceptable safety as well as efficacy, was moved into a phase 3 program. With the INSPIRE trial, which is our phase 3 riboglucosidase switchover studies, the goal we're to look for improvements in endurance and respiratory function in patients that are already on ERT. Currently, 24 patients have been enrolled, and we're looking to add about another 70. That study has a sleep sub-study as well, which I'll talk a little bit about. There's also a 701-201. The numbers are less important. It's our transdiaphragmatic study, which Dr. Smith in Florida is one of our investigators for. The idea is here to see whether or not we can improve respiratory muscle strength by examining the diaphragm directly through the phrenic nerve stimulation that she showed us yesterday. Currently, we had 5 patients, there it is, enrolled, and we're looking to increase that to about 15. There's also a global observational study designed to monitor the endurance and respiratory function in treatment naive as well as those that are already on ERT. There are about 10 patients in that study, and we're looking to get up to 100. What are the goals of our phase 3 program? Well, they're sort of 3 buckets. One is respiratory muscle strength, examining MIP, maximum inspiratory pressure, MEP, expiratory pressure, FVC, as well as sleep disordered breathing. The other is endurance, looking at 6-minute walk test. And finally, quality of life, looking at patient-reported outcomes, both specific for Pompe and for respiratory measures. The trial design for INSPIRE, the phase 3, is rather simple. It's a switch study, so patients remain on their ERT until they are switched over to riboglucosidase alpha, and there's no interruption in therapy. Patients must be on ERT for at least a year, able to walk between 75 and 500 meters, and have some compromise in their respiratory status as defined by reduction in MIP or FVC. The primary endpoint is looking at maximum inspiratory pressure, with key secondary endpoints looking at 6-minute walk test, FVC, and MEP. Of course, safety is a major component of any clinical study. We're looking at antibody formations, both to the enzyme itself, as well as to IGF-1 and IGF-2, adverse events, as well as looking at blood glucose and vital signs. Here's the schematic of the study design. So patients that have been on ERT for 48 weeks are then switched, again without interruption in therapy, over to riboglucosidase alpha at 20 mg per kg every other week. Assessments are performed every 6 weeks for 24 weeks with the opportunity to enroll into an extension phase. And patients are asked to enroll into a sleep sub-study. This is if they choose to, and also in the 701-201 study, which is that transdiaphragmatic pressure study. The sleep study we think is particularly important given the impact of diaphragmatic dysfunction on sleep. And so the idea here is to look for improvements of sleep disordered breathing with riboglucosidase alpha treatment. And so subjects that are on the treatment in 301 or the INSPIRE trial are asked to enroll into that study. And it is an in-center sleep study, and that's particularly important so that we can measure the actual time of sleep, and we can look at REM sleep, rapid eye movement sleep. And rapid eye movement sleep is, if you ever seeing one of your children sleeping or loved ones, you sometimes see their eyes sort of fluttering, and that is the time when actually diaphragmatic weakness may be at its greatest. So it's important to look for that period of time specifically. So in-center sleep studies are particularly useful here. We currently have 29 sites in 10 countries worldwide, in the US, the EU. You'll see many investigators and clinical sites that you know. And we're looking to add another 10 globally. That is my last slide, so it's perfect timing. I want to thank everybody here, particularly, again, the investigators and all the participants. And should you have any questions, feel free to reach out to me. I'm around, you see what I look like, and I'll ask the Biomarin staff that are here just to stand up, so in case you have any questions, feel free to some over there, and there's a lot of us. Well, thank you very much.
Very much. I started to worry and you ended. Fantastic.
I read your mind.
Thanks a lot. The next presentation is by Dr. Susan Sparks from Genzyme Company, and she's Medical Director, Genetic Diseases. So the floor is yours. And the slide is, you recognize?
Yeah.
That one.
Excellent. So I want to thank the organizers and the House family and Tiffany in particular, as well as the AMDA for inviting me to this conference. I have learned a ton and have been very honored to be amongst both the speakers as well as participants of this great conference. So I recently joined Genzyme as a medical director in the US for the Pompe program. And I'm a clinical geneticist by training and have been in the clinic for over 10 years prior to joining almost a year ago. One other thing, I wanted to tell you about this cute little girl on the top of our slide here. This is Sol. She is a 3-year-old little girl from Argentina who was diagnosed with Pompe as an infant, and she is a lovely child who brings a smile to the clinic every day, or every time she visits, I'm sorry. She enjoys school and obviously dance. So I'm going to provide an update of Genzyme. As a lot of you in the room know, that Genzyme is part of a larger company, Sanofi. Genzyme was founded about almost 35 years ago and focused on rare disease. We recently joined the Sanofi organization, which is a large organization, which has increased our ability to spread internationally, as well as introduce more patient-centric care into the therapeutic areas within Sanofi. So you know that there's a long history of discovering and developing therapies for patients of rare diseases, including Pompe disease, and we have a strong partnering with the patient community, with investigators who are caring for patients of these rare diseases as well. So this will be an update. It's not intended for any study recruitment here. So, as you know, Genzyme has a long history of developing enzyme replacement therapy, and we have a goal of looking at the unmet need in the rare disease community in developing therapeutics where there was none before. But in addition, there is still an unmet medical need, as we know, and we don't rely strictly on our therapies that have been developed in the past. So I'm going to talk about the Neo-GAA development program, and it's kind of helpful to go after Biomarin as we talk about a next generation of enzyme replacement therapy. So Neo-GAA is a recombinant GAA enzyme. The figure here is the current therapy, and you can notice these green things are carbohydrates or sugars. So through a complex manufacturing process, Neo-GAA has many more of these carbohydrate structures, which allows it to bind more efficiently to the mannose-6-phosphate receptor, which we've talked about in muscle cells. So earlier this year, we completed a phase 1, phase 2 study with Neo-GAA and are currently entering a phase 3 development. We are reviewing our data from that study and the proposed phase 3 clinical development strategy with regulatory agencies and developing that protocol by the end of this year. We anticipate starting the phase 3 clinical trial beginning in the first half of 2016 in late onset Pompe disease. But in addition to enzyme replacement therapy, we work closely with the patient organizations and recognize that there are many things that we need to learn about this disorder still. So we have been working very closely to develop a patient-reported outcome tool to better understand the experience of patients who are living with late-onset Pompe disease, to make sure that we are looking at those experiences of the patients as well. This research also might help identify and highlight the significant impacts that Pompe disease has on both the patient and their family. So in order to do this, there is a complex process involved, and the first stage involves literature review. And that literature review is what does the professional literature tell us about the symptoms and impacts that are relevant to the patient experience, and what does the literature reveal about how the patient experience has been measured in the past. So looking at current patient-reported outcome tools. We then pursued interviewing physicians who are experts in treating Pompe disease at what signs and symptoms and impacts are the most meaningful to them, what they feel might be the key areas to target. As well as looking at how do those, or interviewing them and asking them how they view the anticipated benefit of a treatment. So this process has been done, and now we're at the point of interviewing patients. How do the patients explain the reality of living with Pompe disease, and how do those patients experience the signs and symptoms as well as the impacts of this disease of what you're living with every day? This has all been completed, and we are actually in the development and testing form. So part of this is qualitative, which means that we're kind of interviewing and getting the process together. But once you have a tool, you actually have to test that tool. So we're looking at, are those existing instruments the best choice for measuring the patient experience? Is there a reason to develop new patient-reported outcomes? And well, it's from all of these interviews with the patients, how well do they understand these new patient-reported outcomes, how the questionnaires, and that's more refining that tool, and how relevant are these areas to the experience of living with Pompe disease. In addition, Genzyme has been partnering with patients like me, which some of you may have heard of this organization, in order to conduct a prospective study in patients with Pompe disease. We want to evaluate the willingness to adopt wearable devices. We are in an age where everybody has Fitbits or some kind of wearable device, very, technologically more savvy than me because I have trouble just with PDFs and PowerPoint, and examine this daily device on measured activity. Overall and longest active period, which might be assessed by a step count or distance, as well as elevation for each patient. And how does the temporal relationship between maybe an enzyme replacement infusion and this device measuring activity? We want to explore the relationship between the patient-reported symptom severity and device-measured activity. So this is in combination with looking at those patient-reported outcome measures. And then evaluating the patient-attributed symptoms that may limit or stop the activity. So on this side, oops, sorry. Oh, I'll go back. On this side of the screen, we have explored the patient experience with a Pompe patient who would join patients like me. This patient would then take a survey to determine the eligibility and baseline status. Patients like me would then send eligible patients a Fitbit and study instructions, and the patient uses the device to log data on the Patients Like Me website over a 6-week period, and then there's an exit survey to provide the status and project feedback. So as you can see, Genzyme is committed to disorders of rare diseases, including Pompe disease, and I would like to thank you again for the opportunity to show you some of our updates and what we're working on going forward.
Thank you. Thank you very much, and we will have the questions afterwards in the panel. Thank you so much. The last speaker in this series is Dr. Wouter Vervecken from Oxyrane, Belgium. So we take this out. Maybe you are more handy than I am.
This is mine. Yep.
15 minutes, no more.
Minutes, not more. Okay.
Otherwise, you are going to miss your plane.
Yeah, I have actually 2 minutes more.
I'm very honored to be.
Make sure to use the mic.
Yeah, I try to.
You're tall, so.
Yeah, I'm very honored to be the last speaker of this conference. I don't believe anything. I need to speak up. Yeah, I'm gonna do your trick over there. So is that working? No, that's not working. Whatever.
Switch that off. Use this one then.
Okay. Okay now? Yep. Yeah, okay. I wanted to say, Arnold, that I don't believe anything about what you were saying, alphabetical order. I think you keep the best for the last, so that's... No, I unfortunately will have to go immediately after this talk because I have a plane to catch and it's really tight. So if you have any questions, you can email me. You'll find my email address on our website. So I will talk. So I'm actually a scientist by training, not a physician, and you will see that in my presentation. It's a little bit science heavy. I hope you don't mind. I'm also coming from another field. I'm trained as a biotechnologist, doing metabolic engineering, and then we found out that we might be able to use these microbes that we were engineering for producing therapeutic proteins. And I must say, it's the first time I'm in such a conference here, and I really, I'm very impressed by the patients, actually, the resilience of the patients. So I'm really, I admire that very much. So let's go into my presentation. So Oxyrane is a small company. We are roughly 36 people. We are focusing on the development of lysosomal storage disease products based on a yeast platform. So these microbes that we have been engineering, I'll go into that later immediately. We're still preclinical stage in our Pompe disease program, and we have investors, VC investors, which are mentioned here below. So, what is our technology? We engineered the yeast to increase the phosphorylation. So, because we've recognized that the current enzyme replacement therapy for Pompe disease has an efficacy that could be improved. Let's put it that way. And how? So we thought about increasing phosphorylation through using yeast, and the yeast is particularly suitable for that because it has a natural mechanism of phosphorylating proteins, and that mechanism is different from mammalian cells, and that allows us actually to put on any glycosylation site of any protein, be it a lysosomal protein, be it a non-lysosomal protein, we can put a phosphorylated glycan. And so we are independent of the 3D structure of the protein or the glycosylation site, and that is the particularity of yeast, and that is what we are using. So our technology is existing out of 3 steps. So the first step is boosting the phosphorylation. We've cloned the genes of the yeast to overexpress these phosphorylating enzymes, this phosphorylating enzyme, and that results in this increased phosphorylation, which you see here in this step 1. We are able to get on roughly 90%, more than 90% of all the glycosylation sites phosphorylated. Second thing is, so yeast does a little bit different phosphorylation than mammalian cells. We won't go into detail about that. But we need to do some enzymatic steps to remove mannoses to expose phosphate, and that is what you see in step 2 and step 3. In step 2 and step 3, and that is allowing us to produce these highly phosphorylated exposed phosphates on the protein. Another thing what we're doing with the GAA is As has been described earlier in this conference, the glucosidase is a protein that is matured as it goes through from the Golgi to the lysosomes, and that maturation has been described in a paper by Moreland, and the picture that I used from this paper, and so the protein is synthesized as this 100 kilodalton form, which is also the enzyme. That is the current enzyme replacement therapy, so myozyme. Then it is processed N-terminally and then C-terminally eventually, and then again at the N-terminal site producing what is called, so here the 110 kilodalton form, the 95 kilodalton form, and then the 76 and the 70 kilodalton form. Remember that these subunits remain attached to the protein, so the protein remains intact. We've been able to make these variants, so we've been able to make the 110 kilodalton form, the 95 kilodalton form, and a mixture of the 76 and the 70 kilodalton form, and a little bit of the 95 kilodalton form as well. And we've looked at the catalytic efficiency and the specific activity of these different products, and what we've seen is what we confirm actually what has been described in the literature, that the 95 and the 76 kilodalton forms have a higher catalytic efficiency and also a higher specific activity towards glycogen. So we have a consistent process for making this variant 2, which is the 95 kilodalton form, and which we call from now on if we refer to the glycoengineered GAA, so the GGAA, that's this form of the protein, and all of the most of the further studies are done with this protein. If we look at uptake, so what is the impact of the increased phosphorylation on the protein is what you see here. So this is a cellular uptake study comparing the 3 different forms of the product that we've been able to make, and with myozyme, and what you see is that the products produced in yeast are better taken up, roughly 20-fold better taken up than myozyme by these Pompe patient fibroblasts. We did a short dosing study in Pompe knockout mice. These were not the mice from Dr. Raben, but the mice developed by Arnold Reuser. And so we did a short study, 4-week study, giving 4 doses at 20 milligram per kilogram. What we saw is a clear trend that the more processed the protein is, the better it is. But we couldn't distinguish. They are not significantly different, statistically significant. We also could show that in the quadriceps, the myozyme product didn't do much of clearing the glycogen, as you can see here. This is a dose escalation study that we did, also a small study, 4 doses, where we could show that our product, if you increase the doses, you get almost close to wild type levels of glycogen. Now I would like to go into these are rather old data, so I would like to go into a study that we did, a 6-month head-to-head comparison study in the same Pompe disease mice that were developed at Erasmus. And this study is a comprehensive study where we did a lot of in-life measurements besides measuring the glycogen storage. So we looked also at grip strength. We did some pulmonary function testing. So we did glycogen clearance from skeletal, cardiac, and diaphragm muscle, and we also did ex vivo muscle strength testing of the extensor digitorum longus. So the study was kind of a big study. We had 15 male and 14 female mice, and so we wanted to do a long-term study. So we treated the mice first with methotrexate to immunotolerize them, so to allow us to do these multiple doses, and that is a protocol that has been described in a publication by Genzyme. So these were the study outcome measures. So we measured cardiac, I mentioned before. These, so we had, I'll go to the next slide because that somehow depicts better how the study was set up. We had to divide the mice into two cohorts, two nearly identical cohorts. We used the males, all the males, for the ex vivo muscle strength testing, and that is that was required because females have hormonal fluctuations, and that messes up the testing actually. And so then the females were all used for the glycogen clearance, and we used all the mice for the in-life measurements such as grip strength and plethysmography. So I'll go into the results. So yeah, the glycogen clearance was typically done by an enzymatic assay after grinding up the tissues and yeah, doing the fluorophoric assay. And these are the results in cardiac muscle. Yeah, maybe important that I didn't mention yet. We did 2 doses of our product at 40 milligram per kilogram and 20 milligram per kilogram, and compared it with 20 milligram per kilogram mice and placebo. In the study, if I go back, maybe, so we only used wild-type animals for wild-type male animals for the ex vivo muscle strength testing, which is a pity actually for the rest of the study, but that was how it was designed. So you don't. It is kind of known that and I think it's also been mentioned here that the heart muscle is treated quite well by current enzyme replacement therapy, and this is also what we see. We see a big difference between placebo and myozyme treated group, and our product is doing equally well. We see at the higher dose that our product is doing better than myozyme. A little bit on the statistics, we built a model and or also corrected for multiple comparisons. I'm just looking at the time, doing fine. If you look at diaphragm glycogen storage, also there we see a little bit the same story. Our product is performing equally well as myozyme, but at 40 milligram per kilogram, this is better than myozyme, and there is a difference with placebo. The difference comes with the quadriceps, so the skeletal muscle. There we clearly see that our product at 20 milligrams per kilogram outperforms myozyme treatment in a statistical significant way, and we also see that myozyme treatment doesn't differentiate from placebo in this study at the 20 milligram per kilogram dose. A little bit on the in vivo strength testing. So this is how we assess the muscle strength of the mice. So you pull on the tail with the mice grab to this T-bar, and then the force is measured when the mice release the T-bar. And we did that for the hind limb and the forelimb muscles. I need to say here the data here in gray are actually from wild-type mice that were not part of the study. They were age-matched, but they were not part of the study, and we didn't do statistics comparing these wild-type mice. But I put them here for comparison, so that we can see what the goal is and how far we're off. We saw a sex effect in this hind limb grip strength, where the female. So we looked at the muscle strength in the after the 6th, the 9th, and the 12th injection, and what you see is that gradually these female mice improve their hind limb strength actually, where and our product at 20 as well as at 40 milligrams per kilogram is is outperforming the current treatment, which is in the red here. And we are approaching these wild type levels in this case. The males are a little bit different. Here we see at the 6th injection already a nice difference between Myozyme and our product, but Myozyme is catching up there in the end. Grip strength of the forelimb, we saw no sex effect, no cohort effect, and therefore also no time effect. So all the data points have been analyzed together, and we see that ERT treatment helps in forelimb grip strength, but is not different for the different treatment levels. A little bit only looking at the time. It's 2 more minutes. Oh, yeah, I'm gonna skip this one. I mean, also pulmonary function testing, we see some difference between our product and Myozyme. I'll go on because these are important data. What we did is we analyzed the muscle performance of the EDL, which is the extensor digitorum longus, which is actually the muscle in your foot to lift your 4 toes, which is the longest muscle in the mouse, and that's the reason why we used it. It's a skeletal muscle, and that has been put into this device, which is a device from Aurora Scientific, where the muscle is conditioned in a way that it's still functional. We looked at a couple of parameters. First thing was the cross-sectional area, and what you see is that the cross-sectional area, which is actually a measure of atrophy, that and here in this part of the study we did have wild-type mice. We see that the cross-sectional area of the placebo mice is really is much lower than the wild-type mice, and also that the products at 40 and at 20 milligram per kilogram start to restore this cross-sectional area, so the atrophy to a large extent, which is then not so much the case for Myozyme. I will go to the next slide first. So this is if you stimulate the muscle for 1 second at 200 hertz. And that is the profile, the typical profile of a Pompe disease EDL muscle. So you have your stimulus, you have a time to the maximal force. There is a force decay which is not very typical for Pompe, for this Pompe mouse muscle, whereas in wild type this is almost a flat line. So you see there is a force decay. There is also an element of relaxation time. If you stop the stimulus, that the relaxation is going slower. And I need to stop, I guess. Basically, what is looking at a little bit of the statistics, I think we're able to restore the wild type levels, or close to wild type levels with our product, where that is still not, where it's definitely not the case for Myozyme. There is also on fatiguing. I think we are also restoring the fatigue levels to the same close at wild type levels, and Myozyme, and we're outperforming. Or our product is outperforming Myozyme on this measurement. We did the diaphragm as well. I know from the study from Barry Byrne that there was nice results from there, but we couldn't make anything out of the diaphragm analysis. Yes,